to share with you some of my own research related to testing adaptive hypotheses, and that is observing a trait in a natural population and then conducting some kind of experiment to determine whether that observed trait is in fact an adaptation or is the product of evolution by natural selection. So one of the things that I study is mating system evolution in plants. How or why species may have evolved to have separate males and females over time, whereas most plants are hermaphrodites, having male and female reproductive parts in the same flower. So one of the systems in that context that I study is called cryptic dioecious. So it's dioecious, having separate males and females, but it's cryptically so it doesn't look dioecious. And one of the species I study that is cryptically dioecious, so this is rare, and one of these I study that is cryptically dioecious is Thalictrum macrostylum. And here we've got pictures of a couple of my images of two flowers. One is a male flower, and you can see that all Thalictrum species are lacking petals. They're apetalous. They've got little sepals that you can't see here. So here is a male flower composed just of stamens, so filament and anther. And anther would be where all the pollen is located. Okay, so really relatively simple flowers, kind of like a pom-pom made of stamens. Over here, the female flower, you see we've got two parts. We've got these stamens with filament and anther, and then here we've got our pistils. At the base of this pistil, you can't really see, would be the ovary in which we've got the ovule, the potential seed. And at the end here, this white part is the stigma. This is where the pollen would land, where it would germinate its pollen tube that grows down through the style into the ovary to fertilize that ovule. All right, so this is what we see in nature. It's possible that, so I've already kind of gotten ahead. I've called these on the right females. It's possible that they're actually hermaphrodites. We've got populations made up of males and hermaphrodites. Let's look a little bit closer. If we look at pollen from a male, it looks like this. There we go. It's aperture. This would be sort of normal pollen. We've got these apertures. This is one of my scanning electron micrographs. The pollen grains are a little bit shrunken in as an artifact of the preservation method for taking this kind of picture. But nevertheless, we've got these thin places in the outer layer of the pollen grain, and this is where the pollen tube is going to push through. Okay, so these pollen grains look like we might expect them to look, and if we do some germination tests, put these pollen grains on a nutrient agar, they do germinate. So here's a pollen grain, here's a pollen tube, here's another pollen grain with a pollen tube. All right, so pollen grains from males germinate. Great. Let's look at the pollen from females. So first off, whoa, no apertures. It's possible that a pollen tube could still come out of the outer layer of the pollen grain without an aperture. So let's investigate. Let's put these inaperturate pollen grains from females onto some nutrient agar. And what we find is that they don't germinate. About 1 in every 200 grains can germinate, right? So more or less no germination. All right, so that's weird. Why would females spend all of the resources? Of course, they're not thinking about this, right? But why would their genetic developmental programs tell them to make these stamens and make all this pollen, this nutrient-rich pollen, if it's not useful in terms of fertilization? So one hypothesis is that stamens in females have some other benefit. So our question is, how could cryptic dioecious be maintained in this species? Why wouldn't we see evolution to having just males and females that don't make stamens? There is variation among females for the number of stamens in a flower. So with genetic variation, we could see evolution acting on that genetic variation. Why don't we? A hypothesis is that sterile stamens somehow indirectly influence fitness. Maybe they enhance pollination, they make pollination more likely or more successful. So let's look at that hypothesis. And this is where our methods of testing evolutionary adaptive hypotheses are going to come in. All right, so our hypothesis is that more stamens leads to more pollen. And we could think of two possible reasons for this. One reason is maybe these stamens make the female flowers more attractive. So if we go back and look at those female flowers, you could imagine that if there were no stamens here, this flower would be really small. It would look not particularly attractive to a pollinator like a bumblebee, who would be attracted to this big, fluffy, pollen-rich flower. Okay, so maybe the stamens attract pollinators, or maybe having these stamens stuck out there around the female flower, around the stigmas, does something with airflow. So as pollen is coming by the wind, it gets kind of caught up in these stamens and is more likely to then settle on the stigma. Okay, so maybe there's some aerodynamic influence here, that it's the structure provided by the stamens that indirectly increases female fitness. All right, so we need to test both of these. We could try to do kind of separate experiments. Like we talked about before, testing all of our hypotheses within a single experiment is a great option if it's possible, because then we're controlling for things like weather and the populations and the particular plants that we tested the hypothesis on. So we can test both these hypotheses at the same time. That's going to be the best thing we can do. So we need to create some variation. I've already told you that there's variation for a number of stamens within flowers, so we don't have to do that. But we need to create some variation in pollinator mode, whether the plants are insect or wind pollinated. The hypothesis about this species is that they are wind pollinated, not insect pollinated, but we need to make sure. I have seen males being visited by bumblebees in the field, so we need to make sure that plants that we're calling just wind pollinated, so we can test hypothesis B. We need to make sure that they really are wind pollinated. So what we're going to do is make little mesh bags to put around the clumps of flowers, the groups of flowers. So it's like a piece of mesh, a square piece of mesh with thread around the outside and two little drawstrings, kind of like a hoodie. So you can pull the drawstrings and it tightens the bag around the group of flowers. So we're going to have some flowers in the bags, and you can see the mesh is fairly large. So it's large enough for pollen grains to fly through, but not so large that pollinators could get in. And then we're going to have some plants that we just leave alone. And you see these are covered in thread because I have each flower as a unit of measurement. And so I've gone in sort of at the start of the experiment as the flowers are about to open and counted the number of stigmas, the number of pistils, and the number of stamens in the flower. So we know what the flowers like to begin with. Then we're going to leave them open for a while, go back, collect the flowers, and then we can assess pollen on the stigmas in the lab. Okay, so we're testing hypothesis B by comparing wind and insect. We're gonna test hypothesis B by just looking at the wind flowers. Okay, and we're going to ask this, make a comparison with one variable being the number of pollen grains received, and the other variable being the number of stamens present in the flower. So we've got two continuous variables, although a mathematician or a statistician would tell you that those are discrete variables because you can't have like 0.25 stamens. Okay, we're going to call them continuous. Never mind what statistics I did. Don't worry, I did it correctly. We need to control for something, which is the possible effect of the bag. So we put the bag on sort of assuming that it's going to create our wind pollinated only and our insect pollinated plus wind pollinated flowers. But it's possible that this bag is doing something weird, like maybe pollen is more likely to land on the flower because it gets in the bag and then it can't get out. Or maybe pollen doesn't get in the bag and it's actually excluding pollens. It's not just excluding pollinators, but it's excluding airborne pollen. So I need to control for the bag. What I did for that is to take microscope slides. And when you're a grad student with very little funding, you get very creative. So this is like a stick that you might use to support a tomato plant or something like that. So like a long rod. And then I wired on here binder clips. And this is a microscope slide. And the microscope slide is coated in grease that you might use to like put around it's for a vacuum, for vacuum pumps anyway. So it's like a grease you would use to lubricate machine parts. It's clear, so it's like a little bit sticky, but not sticky. It's kind of like Vaseline, but a little bit thicker. So spread that on the slide, and then pollen's going to stick to that is the idea. And so we've got some slides open, just like our open flowers, and some slides in bags. And then these stakes are set all throughout the population. Paying attention to how far apart the plants tend to be and sort of mimicking those distances. So these slides throughout the population, I've got some slides facing up and some slides facing sideways just to get a variety. And then we're going to be able to say, we'll count the pollen on the Vaseline, on the grease, and ask whether there's a different amount of pollen between open and bagged slides. Our assumption is that slides on the same stick are equally likely to receive the same amount of pollen, and I've controlled for the height. So sometimes the bagged slides are lower, sometimes the bagged slides are higher. All right. So we're comparing slides in the bag to slides outside of the bag to see if specifically there's an effect of the bag, and there's not. So good job. We can ignore the possibility that the bag is having some influence apart from excluding pollinators. Okay. What data do we need to collect? So our adaptive hypothesis has something to do with fitness. And so we need to collect some data that have something to do with fitness that are going to allow us to assess whether females who have lots of stamens have higher fitness than females who have not lots of stamens. So we should collect some data that's related to fitness. Well, this is pretty easy because we're thinking about pollen, and pollen is one of the pieces of fertilization, carrying the sperm cells. So we should measure the amount of pollen that gets collected on each stigma. So we're going to take flowers away, and we're going to count the number of pollen grains on the stigma under a microscope. Why not? Let's be thorough. This is your dissertation. Let's collect another measure of fitness. Let's look at seed set. So let's leave some flowers out in the field. So this is why it's really important that I put a stake for each plant, and then I've got each flower identified by thread. And you're going to do lots and lots because deer are going to come through and accidentally eat some, and you're going to be really disappointed when you go back. So you got to mark lots of flowers. And so you can see that I've got a flower here with the pink thread around it. So I know exactly which flower this is. I know how many stamens this one had to begin with. And I've got one here with blue, one with white. So I know exactly which flower is which and how many stamens it had to begin with, how many stigmas it had to begin with. And each of these is a single fruit, a single seed inside. This brown part is the stigma. So I can then count for this pink flower out of all the possible fruits, out of all of the pistils, how many of them became seeds. So then that's our measure of seed set, like of the potential seeds, how many seeds did we actually get? What portion of the potential was met here? Assuming, of course, that this has something to do with how much pollen is received. It could have something to do with the female resources available to females. So if a female doesn't have much nutrient resource or photosynthetic resource, then she might not make all of her seeds, might not have enough resource to make all of the seeds. This is why we sample lots of females, lots of flowers. We can control for the effects of the particular female in our statistics. So then we can more or less assume that seed set is a measure of how much pollen you get. Okay, so we've got two measures of fitness across all of our flowers on all of our females in multiple populations. I didn't tell you that. We've got two populations. Oh, this is a fine point. I also controlled for the possibility of pollen limitation that maybe females don't make lots of seeds because they just don't get a lot of pollen. Like there's just not enough pollen to go around. So I did some additional plants where I added pollen. So in that case, the only reason that the seeds wouldn't mature would be if there was some resource problem with the females or some other issue, not that she just didn't get enough pollen. And then so we would compare seed set in the non-hand pollinated to seed set in the hand pollinated, ignoring stamens. That's not what we're looking at here, to make sure that there's no pollen limitation. Okay, that's a finer point. If you want to know more about that, you can read my paper. I'll link it in the comments. Okay, so here are the data. First things first, females are not significantly insect pollinated. We didn't see a difference in the amount of pollen received between open pollinated and wind pollinated flowers. So open pollinated would be no bag, light gray, wind pollinated only in the bag, no insects getting in there, dark gray. I know this bar is a little bit higher, but the error bars are wide, and there's no significant difference between these two bars at one of our populations or our other population. P-value greater than 0.05, no significant differences or degrees of freedom here. Sample size above the error bars. Okay, so one cool point is that this study is the first known study, I haven't read any other studies, that look at cryptic dioecious in a species that's exclusively wind pollinated. Usually we have some insect pollination, so no insect pollination here. Let's then investigate our other hypothesis, our aerodynamic influence hypothesis. Okay, actually, before we do that, let's look again at the open pollinated flowers. So these are potentially insect pollinated flowers, although we see from the previous figure that there's no, doesn't look like insects are visiting. It doesn't look like there's a difference between the amount of pollen you get if you're open, not in a bag, or closed in a bag. But let's look at our open pollinated flowers. And we've got two sites here. Circles are one of the sites, triangles the other site. We've got number of stamens in the flower on the x-axis in both cases. And let me scoot this over. Our first measure of fitness is on the top. Number of grains received, and B, proportion seed set, the amount of the potential seeds that are actually turned into seeds. And there's no significant relationship between either of these measures of fitness and stamen number. Let me rephrase. At one of our sites, there's no significant relationship. At the White Oak WO site, there's no relationship in either one. At neither site is there a relationship with seed set, but at the other site, MC Moores Creek, there is a positive relationship between the number of stamens you have and the number of grains you receive, but it's not translated into seed set. So you might get more pollen grains, but it's not increasing your fitness. And that's what we really care about, is an increase in fitness. So even though you get more pollen grains, it's not increasing fitness. Now, it could be that getting more pollen grains means that you're more likely to get a pollen grain with good genes that's more competitive. And so even if you're not having more seeds, maybe you're having seeds with higher fitness. So it's possible that there is a benefit. It's a higher point beyond the scope of this class, so it's possible. So overall, summary, for open pollinated flowers, sterile stamens might increase the amount of pollen you receive. The Moores Creek site is denser, there's a lot more plants there, a lot of pollinators that are visiting other flowering plants, so maybe you're just kind of more likely to get a casual bumblebee here and there. But no apparent benefit to fitness. No increase in seed set by having lots of stamens. So again, why are females doing this, putting so much resource in? We don't know. Indirectly then, so this doesn't support our pollinator attraction hypothesis. So indirectly, it's support for there being a genetic constraint, a limitation in the amount of genetic variation for stamen number, so that natural selection can't grab onto stamen number and reduce it in females. Maybe because there's a between sex correlation, and so selection on females to reduce stamen number reduces stamen number in her sons, which would be bad for them. So indirectly bad for her and her genes. So there could be some genetic constraints that limit the ability for natural selection to act. Okay, here we are, our last figure for looking at flowers where pollinators were excluded. So flowers in the bags, and we're just looking at the number of grains received. And among our two sites, no difference in stamen number. So this doesn't support our aerodynamic influence hypothesis that there might be something that's going on with the flower that causes pollen to get more likely to sort of deposit it on the stigma, like slows the air down. We see things like that. We see aerodynamic structural interactions in other species, especially there's been some cool research in pines with pine cones and pine pollen, but we don't see that influence here. So sterile stamens do not appear to increase receipt of pollen floating in the air. All right, so we went into little bits of detail about some evolutionary things we haven't talked about yet in class. Also, another example of ways to test an adaptive hypothesis. And this one is kind of a mix of our experimental manipulation and our observational. So we observed stamen number. We didn't manipulate stamen number. I did a separate study where I manipulated stamen number by pulling some off. So we didn't manipulate stamen number, but we did manipulate availability of insect pollinators. So this is really common. We set it up like there are manipulative experiments and there are observational experiments. But there are a lot of experiments that manipulate some variables while leaving other variables observed. Okay, so the key is that we have to create variation in the traits that we want to observe. And sometimes there already is variation, like there is for stamen number in our population, and sometimes there's not. Or sometimes there might be variation, but it's hard for us to get a clear picture of that. And so we can impose that variation, like no insects and yes insects. All right, thanks for watching.